Okay, welcome here. We've got the lecture number 2 for marine invertebrate biodiversity. I'm going to pick up where I left off with classification schemes and specifically talk about cladistics. So when we left last time, we finished up with traditional Linnaean systematics, and we made the point that an issue with traditional systematics is that it requires distinguishing between homologs, or species with similar characters that are of shared evolutionary origin, with those that are convergent from similar evolutionary or environmental pressures. And what this requires to make that distinction is taxonomists with extensive skill and experience with the organisms in question, so that they can make those calls of homologous versus convergent features. And the other issue with this is it's not necessarily a distinguishing aspects that are based on scientific truth, but rather that this is really an attempt to rationalize nature and organize nature in groups that may not necessarily be consistent with their evolutionary origin. And so what we really want in a classification system is one that reflects degrees of evolutionary relatedness, so species that are very closely related or taxa that are very closely related, with ones that are more distantly related or unrelated. And so when we organize this way, all members of a taxonomic group are going to be descended from some ancestral species, and these are going to be more closely related to themselves than to other taxonomic groups. So this is sort of our ideal classification system. And when we look at phylogenetic systematics or cladistics, that's really the goal here. So we're talking about a system, when we talk about cladistics, is one that deals with evolutionary relationships among organisms, is really going to drive this classification scheme. So it's really based on the evolutionary relationships among organisms, and it's based on an idea of Willy Hennig, who was a German entomologist working in the 50s, and he argued that classification systems should really reflect ancestor-descendant relationships rather than similarities. So, so shared morphological, in particular, similarities are less important in classification than our ancestor-descendant relationships. So the evolutionary similarity is really what matters, the evolutionary relationship, not necessarily their seeming similarity. And so one thing that Hennig said is this idea of thinking about these relationships in terms of branching diagrams or trees. And so he said, let's connect all of life together in a branching diagram, and every point represents two species branching off. So this is really where we get the idea of evolutionary relationships in the form of trees. And so Hennig's branching diagrams are called cladograms. And you've seen these, no doubt, in your training thus far, where a clade is a branch. So a clade is a branch. So if we look at a simple cladogram of 3 taxa, A, B, and C, these are our various taxa, taxa A, B, and C, and then this point right here is what's termed a node. So this is a genetic divergence from a common ancestor. So a genetic divergence from a common ancestor is our node. So we've got a node right here where the lineage that leads to A and B breaks off, and from the lineage that leads to C. And then there's another node here where we've got an ancestor that diverges into A and B. So until recently, evolutionary relatedness was really based primarily on anatomy, morphology, and to some extent physiology. And so this was originally based on anatomy and morphology. So the physical structure of the organism, their body plan, and aspects of their morphological organization. To some extent, physiology as well. Now, molecular tools are routinely used to look at evolutionary relationships among organisms based on genetic similarity, as you're no doubt familiar with. Often this conflicts with previous studies, and we'll discuss a few papers in this course that use molecular tools to try to assess phylogenetic relationships among the living invertebrates and compare that with morphological studies. So we'll discuss this this potential conflict as we move through the course. What I'd like to do is introduce some terminology. Before I do, I'm actually going to go back to the previous slide. Let's go back here and let's just erase this. And what I'd like to do is draw up a cladogram that we're going to then reference as we go through the rest of the lecture. What I want to do is introduce you to some terminology in dealing with phylogenetic systematics and cladistics. But what I'd like to do to start with is draw up a cladogram that we can kind of come back to. I'm going to make a couple comments about it, then we're going to define some of these terms. So let's draw out a nice big cladogram here. And I'm just going to have different taxa branch off and just give them or letter designations here. So we've got A, B, C, D, and let's actually modify this just a hair. And at this point, let's have this go right here to E, and then let's do something like this with a dash line, G, and then let's do a dash line to F. So in this cladogram, and I'm going to draw in. So with this dash line I just drew, and I just did some what I'm going to term future evolution. So if you project forward, these solid lines are the true cladogram, and then if we predict into the future, these are future evolutions. And so if we look and we draw some specific characters we can map onto this cladogram. I'm just drawing them in here as solid squares. And this annotation style is going to match up with annotation on a cladogram that I'm going to give you in class. And so if we just draw in these solid squares throughout the cladogram until we get here, let's draw in an open square. And so what these squares are apomorphic characters. So apomorphic characters, and in a bit I'm going to define synapomorphies as an apomorphic character that defines a clade. So for example, this one right here is actually a synapomorphy, because it's an apomorphic character that defines a clade. And so these apomorphic characters are used to construct the relationships in the cladogram. So they're used to construct the relationships in the cladogram. So we're not representing time here, just common ancestry. So this doesn't reflect the time it took for a lineage to break off, rather just reflecting common ancestry. And I'm just going to label each of these apomorphic characters with a small letter. And if there are multiples, we could just do D1, D2, D3, something like that. Call this E. And so what these could be, these could be, let's say, their gills or their wings, or there's some structure that we could then map on this cladogram, or some gene. Okay, so this is just a schematic a cladogram that we can use to talk about some terminology with. So for terminology, again, I mentioned the term clade. When we talk about a clade, we're just talking about a branch. So a branch or a group of organisms that includes the most recent common ancestor, so a group of organisms that includes the most recent common ancestor and all of the descendants of that ancestor. And so what this really represents, a clade or a branch, is going to be what I'm going to term a monophyletic group. And I'll mention that again as opposed to a paraphyletic group as we go forward. So a monophyletic group is going to be an ancestor and all of its descendants. And so a clade really represents that. So a clade is going to be a group of organisms including the most recent common ancestor and all the descendants. If you look at table 2.2 in your text, it's going to show brief definitions for these terminologies that I'm putting up here. So you can always go back to table 2.2 in volume in edition 6 of the Pechenik book, and refresh yourself with these terminologies. So if we look at term 2, again, this is another term I've already mentioned today, and this is node. And so when we talk about a node, we're talking about a branching point in a cladogram. So a branching point in a cladogram, which is going to represent the common ancestor at the time of a divergence. Time of a divergence in two or more lineages. And these are just these branching points in the cladogram, as I mentioned and demonstrated before. So another term is a sister group or a sister taxa. So sister group or sister taxa. So for this term, we've got two groups that are descended from the same immediate common ancestor. So we've got two groups that are descended from the same immediate common ancestor. And so another way to think about this is two groups on both sides of a node. So these are the groups on both sides of a node. And these would be your sister taxa. So in that cladogram I just mentioned, one example is E is a sister to D. So if you go back to that cladogram that I drew up before, E will be a sister to D. D plus E is a sister to C. C plus D plus E are sister taxa to B, and then the next, of course, would be B plus C plus D plus E would be sister to A. And so, again, the groups on both sides of a node are sister taxa to each other. So if we return back to the idea of a monophyletic group, and define that again. So a monophyletic group, as I mentioned before, is a clade consisting of an ancestor and all of its descendants. Let's just erase this. So a clade consisting of an ancestor and all of its descendants. So this is a single evolutionary lineage. So an ancestor and all of its descendants, a single evolutionary lineage. So in other words, when we talk about a monophyletic group, we're talking about a group whose a group of species or taxa whose members are related to one another through a unique history of descent with modification from a common ancestor. So this group of individual or group of taxa that are related to each other through a unique history of descent with modification from a common ancestor. So that's a monophyletic group, an ancestor and all of its descendants. We can contrast that with a paraphyletic group. So we contrast that with a paraphyletic group. In this case, we've got a group in which, WCH, in which the member species, or member taxa, are all descendants of a common ancestor, as we had with a monophyletic group. So we have a group in which the member taxa are all descendants of a common ancestor, but this group that we're delineating doesn't contain all the species that were descended from that common ancestor, but which does not contain all descendants, or all taxa, all descendants of the common ancestor. So we've got a group whose members are all descendants of a common ancestor, but that grouping that we've delineated doesn't include all of the descendants of a common ancestor. And so one good example is invertebrates, right? So invertebrates they're a group which are related by their modification from a common ancestor, but by designating the invertebrates, you exclude the vertebrates, which share a common ancestors with the invertebrates. And so by excluding those vertebrates, you create a paraphyletic group. So if we look at that and sort of draw it out, and if we just, so our example here is a monophyletic versus a paraphyletic group. And we can draw this out this way. So if we just have this grouping of A going into, let's say, species B, and then we've got species C and D coming from species B, and then if we branch off here and let's say we get species E and species G and species H and species I, and if we group off and we say this group right here, as opposed to, let's give it a different color, let's use red, how about that? Right, so if we look at these two groupings, this grouping here is paraphyletic. And it's paraphyletic because it includes the common ancestor A, but it doesn't include all of the descendants of that common ancestor. So it excludes B, C, and D. If we look at this group here, I'm going to switch back to the black. So this would be a monophyletic group. It's a monophyletic group because it includes a common ancestor, B, and all of its descendants, C and D. It doesn't necessarily go back as early as possible in the cladogram, but it does include a common ancestor and all of its descendants. So B is the common ancestor and C and D are the descendants. So let's keep going on here, and we've got apomorphies. Again, I mentioned apomorphic characters, or apomorphies. These are derived advanced characters. And again, these are characters that are going to make up a cladogram. These can be a lot of different things. We often think of them as morphological when we construct morphologically based cladograms, but they can be morphological. They could be developmental characteristics. They could be molecular characteristics. Things like amino acid sequences to phenotypic expression of characters. All of that can be included as apomorphic characters and used to construct a cladogram. And so again, in the original cladogram I drew up, the lowercase a, b, c, d, 1, d2, d3, and e would be representative examples of apomorphies that were used to construct that cladogram. I also mentioned the term synapomorphy. And so I use the term synapomorphy. So the term synapomorphy or synapomorphies is used when characters distinguish between branches and define a group. And so what you're going to see is if you have a synapomorphic character, it's going to be found in sister groups that share a common ancestor with that character, unless that character has disappeared. And so that apomorphic character A in the cladogram defines clade A, and so you'll see that as a synapomorphy in the cladogram that I drew, or in typical cladistics. And so we often think about synapomorphies because they define clades or they define branches as a more specific way to think about apomorphic characters. And we've also got plesiomorphies. Plesiomorphy is an ancestral character. An ancestral character. So a character is present but doesn't distinguish or define a clade. So a character is present but does not distinguish or define a clade. And so you can contrast this with synapomorphies, which define clades. So you have synapomorphies and plesiomorphies. And I'll give you an example of this right here. So if we look at synapomorphies versus plesiomorphies, right? And so I'm just going to draw two cladograms and show you how a character can go from being a synapomorphy to being a plesiomorphy. And so if we draw a cladogram at this scale, where we've got, let's just say, bacteria, and we've got plants, and we've got metazoa as our major groups. So let's draw on some characters here. Let's say we've got organelles and sexual reproduction, as characters in the plants and the metazoans, but lacking in the bacteria. And then we've got chloroplasts as a character of the plants, and then we've got a flagella as characters of metazoans that are lacking in bacteria and plants. And so in this case, we've got a flagellum or flagella that are synapomorphic characters. Let's draw a different cladogram, in which case we've got sponges. So these are all metazoans. Ctenophores. Arthropods. Echinoderms. And vertebrates. So all of these are taxa that we'll be dealing with. And in this case, let's draw in a flagellum. So flagella, we've got organs, we've got a one-way gut, got an exoskeleton. Oh, let's put in a vertebrae, backbones. So let's put in these structures here. And so in this case, we went from flagella being a synapomorphic character here to being a plesiomorphic character. All just based on how the actual cladogram was structured, right? So all based on how the cladogram was structured. Let's skip that. So one problem is with phylogenetic systematics and cladistics is that they're made by assembling clades or branches to achieve the smallest number of evolutionary steps. And this is a technique called parsimony. So parsimony is going to be assembling cladograms by minimizing the number of evolutionary steps required to get the traits that you're analyzing among taxa. So this is a technique of parsimony, but the problem is kind of a question. Can we assume that animal evolution moved from one form to another in the most direct way possible? So really, did evolution move in the most direct way possible? And perhaps this is something that we can discuss as we discuss this material of cladistics when we meet next time. So an interesting thing to think about: is parsimony an issue, or is it a blessing or a curse? Okay, so that's it for this coverage of lecture material for cladistics. I'm going to stop here, and that's it.